Okay, good morning. We're going to continue with methods in complex systems. I'm going to continue where I left off last time in terms of discussing fractals. And I have some announcements, which I'll make later. So let me sort of reorient ourselves to where we are. And you see that I'm too jet-lagged even to say that I just flew in from LA, and boy, are my arms tired. So I haven't done that. So as you can see, I got another T-shirt. Which if I get out of the way, we can, okay. So, thanks. Where we are is I'm giving a review of fractals. And let me state again why I'm doing this. Because we've seen so far in the class where we have Gaussian statistics, which is the usual stuff, and then proceeded to a type which we called non-parametric statistics. So the Gaussian is characterized by all the usual stuff like z-scores and t-tests and analysis of variance. And to non-parametric, which include things like rank sum tests and similar things. And now we're getting on to fractals, which I almost misspelled, so things really are bad. In each of these, what we're doing is lightening up on the assumptions that are involved in what goes on, in how we expect the data to behave. So the Gaussian is the most restrictive assumptions. The non-parametric doesn't assume it's Gaussian, but assumes the moments exist. And now we're dealing with the fractals, where we have no moments, where the distribution itself is not normalizable, which in a technical sense means the integral of the probability density function is not equal to 1, which is sort of makes it a very strange probability function. So let me say where we left off last time in fractals, and then we'll continue at that point. Where we left off last time. Well, what? What? Oh, sorry. No, that's okay.
Normalizable took a lot to...
Okay, sorry. And then I can rewrite this in probably even a more accurate way. So if we have a probability density function that depends on a variable x, and we integrate over x, typically that equals 1, and that's how we define probability. But in this particular case, it's not equal to 1. And so our usual definitions of probability density functions are kind of odd in this case. And some of the exactly how we handle things is not completely clear here. Okay? Okay. So where we left off last time, we said that fractals have kind of a defining probably, which I called self-similarity, which means that we have some object, and if we look at a little piece of that object, the little piece has some resemblance to the whole object. Now, in real biological systems, what we're really dealing with is what I called statistical self-similarity. Because it's not true that things are exact copies of their larger pieces, but they're kind of approximate copies. And the definition of statistical self-similarity is that some statistical property, for example, is measured by the probability density function. So this is the number of pieces of a given size of an object, x. So this is the number of pieces that an object has of size x. And if the probability density function has a certain form, when x is large. For example, if x runs from 1 to 100 centimeters, then self-similarity means that the probability density function measured when x is small, for example, it runs from 1 to 100 micrometers. And again, so this is the distribution of the big pieces, and this is the number of pieces that have size x for the small pieces. Then these two distribution functions have the same form, so that they're similar in shape. And that's the definition of statistical self-similarity. Now this is a little bit of an unusual starting point for fractals. People would normally start with the Hausdorff-Besicovitch dimension and talk in terms of dimensions. In a certain sense, I think, and some other people think, that, or at least one other person that I know, was a mathematician, so that would be the accurate way of saying it for him, that in some sense this idea of self-similarity is a more basic notion than the concept of dimension. And so I'm starting here from this notion of self-similarity in defining fractal and deriving the other properties from it, rather than in terms of dimension, which in the mathematics literature has been historically the direction from which this has been approached. What that means is, starting from this notion of a fractal, we were then led to this idea of scaling, which we don't have in either the Gaussian or the non-parametric statistics. So for example, if we have a coastline, this is sea, I guess is blue, and this is land. Land, I guess, should be brown. That if we enlarge a piece of the coastline, that it still looks just as wiggly, no matter how much we enlarge it. And the scaling property here is that if we measure the length of the coastline, that is, we go to smaller and smaller scales. So for example, if this is on a scale of 100 kilometers, see we got very scientific here, instead of miles, and 10 kilometers and 1 kilometer, which I guess should just be in the opposite. Let me make this 100, 10, and 1, because I've blown it up, so this is the highest resolution. That as we go to finer and finer scale, the length we get gets longer and longer. So in an article entitled, "What is the Length of the Coastline of Britain?" And the answer is there is no length. There's no fixed length. That the length depends on the resolution at which we measure it. And in particular, we found that if we plot the log of the length against the log of the scale at which it's measured, then we get a straight line on this log-log plot. So as we go to smaller and smaller scales, we catch more and more bays and peninsulas, and so the length we get for the coastline keeps increasing. Now this property is very, very different than what we're used to seeing in terms of doing measurements, because we're used to really doing a measurement and calculating a mean value and getting what the answer is. And so here we have a system where the mean is meaningless, to use an expression that's in the title of a couple of my papers, but really comes from Hausdorff-Besicovitch here. So we're really dealing with a situation where the mean is meaningless, where the mean depends on the resolution at which it's measured. And this dependency of how a measured property depends on the scale, I've called, in other people, the scaling relationship. And this is where we left off 2 weeks ago. I was giving examples of scaling relationships. And let me give you, so the suspense doesn't kill you, what we're going to cover next, so the ship does sink at the end of the movie. We've started off with this concept of self-similarity. And from that, we now have this concept of scaling. And I'll give more examples, biological examples and other examples of scaling. And then we'll see where this leads in, we'll use dimension to quantify both of these concepts. Remember, we have no mean anymore. So we'll see that the dimension, in some sense, will replace moments like the mean and the variance. So now we're dealing with a different kind of statistics where the dimension will be the essential unit that we're going to be talking about. And we'll see as explicitly as possible, especially through some examples, how we handle the statistics of that. So that's the direction we're going to go in. And when I push the magic buttons, we'll get back to here. So over here we have from Richardson, the person who did this, I don't know if I described him last time. Richardson was a physicist. Did I talk about Richardson last time? Okay. Was a physicist who did this, among other things, and studied turbulence. And did this because he was interested in the causes of armed conflicts. And what he's plotted here is the length of the coastline against the size of the resolution in which he measured it. And if you could switch to the camera for just a moment, guys. It's okay. The way Richardson measured the coastline was with an instrument that not everybody seems to know the name of, which is called a divider and looks like this, and has got two metal points and comes in a nice box with usually green velvet, or sometimes black velvet. And you can set this at different distances, and then you can walk around the coastline. So what Richardson did was use a divider to walk around the coastline. As you take smaller walks around the coastline, you catch more of the peninsula, more of the bays and peninsulas, and so the length gets longer. And that's what he's done here. So this I showed last time, but we're starting here because it's a very classic example, and we can see that the coastline depends on the resolution at which it's measured. And biological example I also gave last time was the scaling of membrane area by Baumgartner, Lohse, and Wiebel in Germany. And this just recalls the data I showed last time. But I'd like to, again, emphasize some points on this. Here we have the membrane area of the inner and outer mitochondrial membrane and the endoplasmic reticulum, which is the place on this cell where there are ribosomes, so that messenger RNA is translated, that is copied from DNA. The messenger RNA is spliced. It gets put into these ribosomes, which then translate the codons into proteins. And these are little bulges that stick on this cell membrane, or in internal membranes, really. And we see here, this is the resolution of the electron micrograph at which it was measured, and this is the measurement of the area. We see here there's a straight line on this log-log plot indicating we have a power law scaling, in this case over maybe 1 or 2 decades. And then the scaling ends. And this is very typical of fractals, that we have some regime where the system is fractal, where we have the scaling, and then either that dimension changes or it's not fractal here. At this resolution, we're really at atomic levels, and we're seeing the atoms in the membrane, so the membrane area is fixed. Now, there's been a controversy recently where David Avner, who's done a lot of work on fractal catalysts and materials, wrote what a number of us think is an incendiary and stupid one-page article in Science about a month ago, where he said that he looked through the literature and found how many decades of scaling, that is, of this sort of scaling, are present in different fractal articles. And they extend only up to 3 or 5 orders of 3 orders of magnitude. And therefore, he said, since it doesn't go on forever, the world isn't fractal, and implied this stuff is useless, which is nonsense. And I think what most of us feel, talking to several people about this article, is that the issue, not everyone feels this way, but I think the strongest feeling that a number of us have is that the real issue is not over what regime the scaling occurs. Or let me say it in a different way, because this is an issue that comes up in a number of different areas in fractals. So let me say it in this way, in that the biological fractals are not equal to the mathematical fractals. So the mathematics is not equal to the biology. And in a certain way, this may seem obvious, but it's not. A lot of the mathematical formalism is developed in a way which doesn't apply to the world that we live in. And by dealing with a lot of real-world problems, it actually helps us to change and develop the new mathematics. So for example, most of the mathematical definitions of dimension we're going to see usually take the limit as the size of things goes to 0. And in the real world, we don't really care about that. We really have a scaling regime which will be finite. So we can see the scaling over a finite regime, and that will allow us to get the dimension. And we don't need the formal mathematical definitions to get at the dimension. Now, that doesn't mean that we cheated. It means that the mathematics has developed materials in a way that don't directly apply to what we need. And in a way, this is good because this gives us a way of reinventing the mathematics, which is in the direction that I'm presenting this, in terms of starting from self-similarity and developing the dimension instead of starting from the dimension. And this has happened frequently in physics a number of times, where, for example, Fourier actually developed Fourier transforms to deal with heat problems. And the mathematics of Fourier transforms are very sloppy, rigorously, as far as rigorous mathematics go, and then eventually were cleaned up. And this mathematics has developed a way that it wanted. And as I said, the problems we face in biology are very different, and we've got to be careful about not making the mistake of applying this blindly to the biology or the physics. And this has also come up in an algorithm that I developed that I'll describe later on box counting, where it was criticized because we didn't take this limit. It wasn't criticized very much, but it was criticized a little bit because we didn't take this limit. And the answer is, you don't need to take this limit. Okay, so this issue of the mathematics not being exactly what we need for the biology, I think the biology and the physics are driving new mathematics. So the point about what David Avner was saying is that what most of us feel in terms of this is that we don't need this to go on forever. This doesn't need to be a mathematical fractal. But if this goes on long enough so that this scaling tells us something about the system, then the fractals have helped us learn something, and then they're useful. Okay, so that's the bottom line. Have the fractals, for example, in terms of the scaling, either been a much better way to characterize the data? Because if the mean doesn't exist, then using the mean is not a helpful thing. So if we can characterize the data with the fractals, or if it can lead us to an understanding, meaning, for example, some hint at the mechanism that underlies the scaling, then they've been useful. And that's what the crucial point is, not whether the scaling goes on for an infinite amount of time or over a very, very large range. It's whether it's a good way to characterize the data and whether it leads us to some understanding or hints about mechanism. And not, so the issue is not that since something is not fractal forever, since it's not always fractal, the idea that that applies that fractals are not useful is, I think, not the case. And so it's odd that David Avner wrote this article, because he should know better. And people like us don't like this article because it affects how other people view the field and can have important political consequences. So let me leave this on long enough, because I think this is a really crucial point, and I think it's an issue, because I'm asked frequently, you know, if you keep going to smaller scale, you eventually get atoms or quantum mechanics, and so this all falls apart. And the answer is yes, but that's not the issue. If we've learned something by going across a number of scales, then it's useful, and that's what the crucial point is. So that's actually very important. And I slid before pushing the button. That's illustrated here. We talked about ion channel kinetics last time. We found that there was a scaling that we could interpret in terms of distribution of energy barriers or how those energy barriers changed in time. And I'm using this to try to understand the structure or the shape in ion channel proteins. I don't remember talking about this, but I must have, so we'll pass over it because I know I was past this point last time. So to give examples of where the scaling relationship has been measured, we've seen in terms of membrane areas, in diameters of airways in the lung, in the size of spaces between endothelial cells in the lung, and in the surface areas of proteins. Maybe that's worth discussing for a moment, because I was reading about this on the airplane. If we have a protein, it actually has a shape, which I've shown here in 2 dimensions, and there are pockets or holes in this shape. One way of measuring the area is by taking a molecule and rolling that molecule over and seeing how long the surface is. You can see that this molecule will catch big pockets, but if there was a very narrow pocket inside the molecule, it wouldn't find it. And so as we roll molecules of different sizes, we get different surface areas, for example, of the protein. When they started to do this, the typical size of a molecule they rolled was about equal to a water molecule. But now we can see that different molecules that are bigger and smaller will have access to different parts of the protein. And people are using this measurement of membrane area to characterize the roughness of molecules, and also to try to see whether some parts of the molecule, for example, might be different from other parts. And this difference in a local fractal dimension might indicate sites, for example, that bind to small molecules and are involved in reactions or things like that. So this is proving to be very, very useful. There's also a scaling of the total size of the molecule. So for example, if you plot mass or volume of molecules versus their mass, that is their molecular weight, you get a certain relationship. If these things were just filled in spheres, and volume would go like radius cubed, or volume would go, since the mass, like proportional to the mass. But what we see is actually the volume goes to the mass to some power, which is not 3, not 1. And so we get on a log-log plot a different slope here. And that tells us something about how compact the proteins are. Proteins collapse. Let me say it in a more technical sense. A protein, when it starts off and comes out of a ribosome, here's a nice blue ribosome, which consists of a number of units, but primarily 2, and messenger RNA comes through here, and the protein is grown out of here. When this so this is a protein. This protein then forms into some 3-dimensional shape. Here it looks like it's crossing itself, but only because I projected it onto a 2-dimensional plane. And it forms a shape. And the question is, how compressed is this shape? That is, the shape might be rather loose, or it could be more compressed. And in fact, you can show from measurements that this shape is quite compressed. If you have a polymer where the units of the polymer like each other more than they like the solvent on the outside, then in the center of the polymer you're going to get a lot of these units, and the structure will be very compressed. In terms of a protein, we say usually the interior is hydrophobic, that is, it doesn't like water, and since there's water on the outside, the interior collapses to a ball. In fact, the dimension here is actually, as we'll see in a moment, is close to 3. It's a little bit less. It's about 2.8. So it's not completely collapsed. On the other hand, if we put the protein in a solution that had a solvent where the units of the protein like the solvent, then what would happen is that this structure would unfold because individual units of the protein could now be more associated with the solvent. And we'll see this leads to a more expanded fractal dimension of something like 5/3, 1.3. So people have also used fractals to look at the configurations of proteins and how they change, and to study the interactions between the solvent and the amino acids of the protein. So that's more about proteins than I ever thought I was going to say. But so that's an example where people have looked at scaling relationships. People have also looked at scaling relationships not only in space but in time. We've seen the kinetics of ion channels. Another example are the rates of chemical reaction limited by diffusion, which I think I talked about last time. But maybe I'll say a few more words again, because there's a lot of work been done on this, and it's I think, interesting. And it also shows how the normal assumptions we make with ideas we normally don't always realize. So let's say we have a chemical reaction where I'll use all the colors I have. A plus B goes to some other third species, which we'll call C. Well, you can write down the kinetics for this chemical reaction. Typically, the way we write this is that the rate of change of A in time is proportional, in this case, to the A, for example. And so the amount of A in time goes down like a single exponential. But although this looks very simple, in fact, the assumption here is that the A's and B's are well mixed. So an A can always find a B in order to interact. And in fact, in the real world, that's not going to be the case. So if I have A's here, there'll be a little inhomogeneity between where the A's and the B's are. So what will happen is that, for example, this reaction may happen, and then a little bit later, maybe these two will find each other. But we'll now create a space here where, if I draw it this way, as more things interact, all of the ones in between, if the C's are here, we'll have places where things have interacted, and then we'll have other regions where there's just a predominance of one form of the other, and so the reaction hasn't happened yet. And then where the reaction will happen now is on the interface only of the A and B regions. So the reaction will be slower in time. And in fact, if we look at the decline of A in time with time, instead of being an exponential, it's going to be much slower because the reactions can only happen on these boundaries. So this assumption of an exponential decay really depends on the fact that we took everything and started infinitely fast and infinitely well at every moment. So all of the chemical kinetics that you've seen in terms of biochemistry and other chemical reactions have this hidden assumption when we write down equations like this that we're mixing things everywhere in space and time infinitely fast, which is a little bit odd. Real chemical reactions don't do that. And this shows an example of just writing down equations and getting so used to writing them down that we don't really remember the assumptions that are involved. And a specific example of this occurs in, I'm not going to be able to produce the details of this off the top of my head, in something called Michaelis-Menten. So let's see if I can spell it something like this. Is that close? Michaelis looks wrong. How do you spell Michaelis? Looks close? Okay. In these sort of chemical reactions, if the dependence of the rate constant of the reaction goes like the concentration of one of the species to some power, like 4, then what we think is going on in the reaction is that 4 A's need to come together in order to participate in the reaction. So for example, A could be chlorine or oxygen or something like that. But it will turn out that, in fact, this does not necessarily tell you that if this reaction is limited by what it says on the computer, which is diffusion, so in reactions limited by diffusion, this is not the case. An example is the one I just gave you, because these areas are not diffusing very fast, so that's why these are staying separate. The reaction is limited by when this A can reach that B, so it's limited by the diffusion time. Then it turns out that this number represents something about how long it takes for the A and B to diffuse to reach each other, and really doesn't tell you about how many A's are involved in the reaction. So this is an example where the fractals have led to a different physical picture of what's going on and a different interpretation in terms of how you handle the data telling you what's going on in the reaction. And there's been a lot of work on diffusion-limited chemical reactions this way. And again, I've done this. And where we left off last time actually was here. It was the washout kinetics of substances in the blood. So let me describe what this means. It's possible, for example, to measure the concentration of substances in the blood. So if I take this, which is my or your arm, and it has blood running through it, arterial blood and venous blood. And we sample these in time, so we make a plot of some substance in the blood versus time. And just for fun, let's make this a log-log plot. This is called washout kinetics. Because it's how the substance disappears in time. And this could be anything from a substance you're given externally to a substance that was produced accidentally in your body because your kidneys stopped working, for example, and then the kidneys have kicked in again and you're watching a decline of urea in time or something else like that. Very often in a plot like this, what they look like is something like this. I didn't bring any with me today, but this is sort of typical. So we have regimes here where we basically have no scaling, where we have a constant value as if it was a Gaussian. And then we have regimes here, so I would say this is, can describe this as not fractal. And we have these other regimes where the washout kinetics obeys a power law scaling, so it is fractal. And let me first of all discuss the interpretation of what it means when it's fractal, and then what it means when we have these combinations of regions of different sorts, because this is very common in a lot of types of experimental data. We can get a fractal region, which apparently is in red, when we have lots of processes going on, either in parallel or in series. So for example, the usual, maybe I can make this clearer by saying the opposite, which is non-fractal process goes on when a compartment in the body consists of just a single volume, and the stuff in that volume just leaks out in time. So when we have one size or one unique volume in the body, one compartment, so each of these pens is one compartment in the body. So the first one was the best one. In the fractal, what we could have is either we have in parallel a lot of compartments of different sizes, and the stuff is leaking out of big compartments and smaller and smaller compartments. Or we could have compartments in parallel where we have nested boxes and smaller compartments inside each other. And the stuff needs to leak out in between all these compartments to get out. So this is in parallel, and this is in series. And physiologically, actually, this seems like a more likely description than this. So these fractal times are times where the stuff in the body is dealing with lots of different compartments, and the non-fractal times are where it's kind of just predominantly leaking out of a compartment at a given spatial size, which therefore corresponds to a given temporal scale, time scale. Now, this characteristic of regions, as I said, within a non-fractal and fractal is very common in a lot of different phenomena. In one of the books I have that discusses geophysical phenomena, there's a wonderful quote where he says that there are just different processes in nature. There are some processes like wind erosion that may extend over many, many scales of water erosion, which I could see yesterday over the Grand Canyon, for example, where there was lots of erosion of different scales. In fact, it was interesting because it was very wide, so I don't know if that means the rivers were very wide at one point, but it seems surprisingly wide to me, and cut pretty deeply, but the width worried me more. So there are processes that extend over many scales, and there are processes like volcanoes, which extend only over one scale. And we have mixtures of processes in any physical or biological system, some of which extend over one scale like these, and other processes that extend over many scales. And the real world consists of mixtures of processes like this. So let me say that. So that in many areas in physics and in biology and chemistry, it's not that we have a single fractal, for example, but we typically have combinations of processes, some of which are local in space or time, meaning they have a single scale, and some extend over many spatial or temporal scales. And this is a very general feature, and I think this is important to keep in mind in analyzing real data. So I think this is an important point from the real-world analysis of real data, that we have both volcanoes and wind. Some of the washout stuff was done by Weiss, who's in Northern Europe, I forget, either Holland or Scandinavia. And we've corresponded a little bit about some of this sort of stuff. It also shows that the work that you do in one field in terms of the ion channel kinetics, which also has these characteristics, can give you insights to how to handle another field. And some of the insights that Weiss had in handling his work, or some of the interpretations, he found some of the work that I did on ion channel kinetics very helpful, where we have basically the same thing. In terms of the ion channels, what we think is that these two different processes, these single scale and multi-scale, may correspond to different structural, different dynamical processes in the channel protein that have those different kinds of characteristics. And this time I push the button first. Oops. Okay, so to summarize what we've learned from scaling is that scaling means that as we change the resolution, the value we measure for a property changes. So this illustrates a fractal that has details at finer and finer scales. And so, for example, if we measure at a resolution of 1 centimeter, we may find that the perimeter of this thing is 5 centimeters. But if we measure it at a smaller resolution, then we may find that the perimeter is somewhat larger, that is at 12 centimeters. And this is really what we mean by scaling. And again, just to bore you to death with the fact, assuming the chair doesn't collapse, is that this is really unusual because we're really not used to having this. We're really used to having a situation where we measure something and get a single value. So this is really surprising. In a way, it's really counter to all the statistics I was developing at the beginning. So it's very important and different from what we're used to seeing. And so the take-home lesson on this is if we plot the logarithm of the measurement against the log of the resolution used to measure it, what we're used to finding is one value. But what we really want now is if we know that the value we get for the measurement depends on the resolution, then what we really need to do is to measure at a number of different resolutions. And what is important to us is how the measurement depends on the resolution, which is indicated by the slope here, and that's going to be the relevant parameter. And in a few minutes or less, we'll see that this slope is a parameter called the fractal dimension. As someone was telling me last week, in some ways this is kind of depressing because it means we have to do a lot more measurements. See, here we only have one dot, and here we have more than one dots. If I was less jet-lagged, I would try to count the dots, but I'm not going to try to do that. But there's certainly more than one. So in some ways, this increases the difficulty we have in the task of characterizing the system. On the other hand, if we have done this, then we have more information about the system than just that one value. So now let me discuss dimension. As I said, the way I'm approaching dimension is as a measure of self-similarity and scaling. So as I said, I started with the concept of self-similarity, showed that that led to a scaling, means that the moments depend on the resolution at which they're measured, and now we're using dimension as a way of quantifying self-similarity and scaling. Yeah?
That last graph, was the line going the right way?
For this one?
The second one,
Yeah, usually we'll see a lot of things like this, but we'll see in a moment too that whether this slope is negative or positive depends on the physical system. Usually we have a situation where as we get to finer values, like in the coastline, we get larger measurements. So more often we'll see a slope in this direction, but we'll see that the slope depends on the nature of the physical measurement and could either be positive or negative. So what the dimension will tell us then is it will tell us as we go to finer resolution how the system changes. In particular, it will tell us how many new pieces we see when we look at finer resolution. So this is the qualitative meaning of the dimension. That the dimension tells us how many new pieces we see when we look at finer resolution. And I have to wait till I see people's pens stop moving since I'm not writing. Right, the whole purpose of me writing is that so I don't get too far ahead of you. So, and you can see how bad a speller I am. Okay. So there are different types of fractal dimension. And what I'm going to do today is discuss three different types of fractal dimension. One called, all right. Let me say it differently. I'm going to discuss three types of dimension. So first of all, we're going to discuss fractal dimension. Then I'll discuss a different dimension called topological dimension, and then a different dimension called embedding dimension. So we're going to discuss these three different dimensions. Let me write down the three overall types over here first. So the three types of dimension are going to be the fractal dimension, the topological dimension, and the embedding dimension. And we'll see each of these has somewhat different properties that will be useful to understand. So the first one of these is the fractal dimension. And there are a number of different types of fractal dimensions, which we'll discuss too. Each of these dimensions tells you something slightly different. And the fractal dimension determines the space-filling properties of an object. So that's the crucial aspect of the fractal dimension, that it tells you about the space-filling properties of an object. And examples of the fractal dimension are the self-similarity dimension, capacity, and the Hausdorff, so there's no O here in the middle, the Hausdorff-Besicovitch dimension. I always feel badly when I misspell Besicovitch. It seems like someone like Liebovitch should at least spell Besicovitch correctly. So apparently I have less problems with Hausdorff. Besicovitch was Russian. Hausdorff was probably German, but I don't remember. And we're going to see examples of each of these. But before we see that, we're going to see this other type of dimension called the topological dimension. And the topological dimension is different than the fractal dimension. The fractal dimension can have any value, an integer or a non-integer, like 1.0 or 2.8376. But the topological dimension can only be an integer, like 1, 2, 3, 0, 1, 2, 3, 4, 5, 6, numbers like that. So the topological dimension has different properties. And it tells us, as we'll see, how points within an object are connected to each other. So it's a different sort of business than the fractal dimension. And I'll discuss two examples of this, one of which is called the covering dimension, and the other of which is called the iterative dimension. And the third sort of dimension, which has less subcategories, is called the embedding dimension. Now, this one can also be an integer or a non-integer, like the fractal dimension. So the embedding dimension is the space that contains an object. So, for example, the space that contains us in this room is three-dimensional. So the embedding dimension here is 3. If I do the shadow of my hand on this thing, which you can see there, not well, but I can see it, that shadow is 2-dimensional. The embedding dimension here is 2-dimensional. So I'm taking my hand and crushing it into a 2-dimensional space. So the embedding dimension here for the shadow of my hand is therefore 2 dimensions. So the embedding dimension is the dimension we put something in. And this can be an integer or non-integer. If I was clever, I could crush my hand into a Sierpinski triangle, which has a dimension that's not 2. And in fact, people make things like Sierpinski triangles. Looking for a picture of a Sierpinski triangle. People make them actually on silicon substrates, which have electrons and holes which interact when they find each other. And so in that case, you have a reaction, in this case, of charges and positive and negative charges wandering on this lattice, and the lattice itself has a fractal dimension. So we can have situations where we have a fractal dimension as the embedding dimension where the system is living. So this is the space that contains an object, or the space where a reaction is happening. Now let's go back to each of these dimensions and describe them in a little bit more detail. Again, let me say where we are. What we're trying to do, since the moments like the mean and variance don't exist anymore, we're trying to develop parameters that characterize the system. So the fractal dimension, in a way, is a replacement for the mean or the variance. So the self-similarity dimension is illustrated here. And it basically says that what we do... Good. It basically says that what we're going to do is take an object and we're going to cut it into m pieces. So we're going to replace each line segment by m line segments. And when we do that, we're going to find n pieces, each of which is geometrically similar to the original object. So I'll say that again. We're going to cut up the object into line segments, each of m line segments, and we're going to see if we have n pieces. Each piece has got to be geometrically similar to the original. When that's the case, then n equals m raised to d, where d is the dimension. So this gives us a dimension. And now we're going to see 3 examples of that. So for example, if I have a line segment and I cut it into thirds, 3 pieces, 1, 2, 3. First of all, see that each of these pieces, this one piece, is geometrically similar, self-similar, to the original piece. So that's why this is called the self-similarity dimension. So each little piece is similar to the whole big piece. So now we cut it up into thirds, 3 line segments, and we see 3 pieces. So 3 equals 3 raised to the first power. That means the dimension of a line is 1. Okay? Now here, I've also cut each line segment into 3 pieces for this object, which is a square area. But when I cut each line segment here into 3 pieces, so this gets 1, 2, 3, 1, 2, 3, now we have 9 little squares. So each of these little squares, again, is self-similar, looks like a smaller copy of the big square. So now 9 equals 3 squared, so that means that the dimension of this thing is 2. So far we found that the dimension of a line is 1, and the dimension of an area is 2. So that shouldn't be a big surprise, but it shows you that this is consistent with the intuitive idea we have of area. So a lot of times we have formal definitions, but it's nice that they should match our intuitive feeling of what the system is. Sometimes the definitions don't do that, and sometimes they do. But you can see why this is called a dimension. And as you might suspect, the next one we're going to deal with is a volume, where you can see, at least in this case, all of the boxes aren't labeled. But in my book, every single box, except the hidden ones that you can't see that behind, are labeled. And so now we have this cube, which is a volume. It's just like as long as games don't appear that you have to get the right answers to, or the computer crashes. We'll see if ultimately that message will crash the machine or not. We have this volume, and now we have 27 little cubes. When we divided each one by a third on this side, we get 27 cubes, which is 3 cubed, so the dimension here is 3. So we've seen that this self-similarity dimension is consistent with our intuitive concepts of a line being one-dimensional, an area being two-dimensional, and a volume being three-dimensional. Now the problem with this is that in the human body and in the physiological processes that we deal with, the little elements are not exact copies of the big elements. So this is not good for handling statistically self-similar objects. It's good for handling geometric or mathematical fractals, but not good for handling our real data. So we need a generalization of this. And one of the I'll talk about 2 of them. The first one is called the capacity. And what we do in the capacity is we take an object and we cover it with things in mathematics which are called balls. The definition of a ball is all the points within a given radius from a central point. And that can be in 1 dimension or 85 dimensions. So they're sort of the ball is a generalization of a sphere. And if we cover our objects of balls using the embedding dimension for the balls, for example, we'll need a certain number of balls to cover the object, that is, to contain all the points of the object. Now, it may be that to make sure every point is contained in the object, the balls will overlap with each other. Okay? So now we've got to be a little tricky. What we really want to say is the minimal covering of balls, the minimum number of balls to cover the object. It's a little tricky how you figure this out, actually, but it can be done. And here we're also taking balls of the same size. So what we do now is we reduce the size of the balls, and we see as we go to smaller and smaller balls how many balls are needed to cover the object. So you can see this is a little like what we were doing before. The balls represent in some ways the scale of the object, and we're seeing as we go to smaller scale how the details of the object change with the scale, in this case, of the covering balls. And again, this will be a minimal covering also. And then what we do, in order to get the dimension, is we say the dimension is equal to the limit as the size of the balls go to 0. See, I said that mathematicians would say this, and indeed right now they have. The limit as the size of the balls goes to 0 of the logarithm of the number of balls at a given size divided by the logarithm of, actually this n shouldn't be here, it should be just log 1 over r. So what I want to do, there's some other words written down below here. Let me explain a little bit more what those words mean and how the capacity is really very, very similar to the self-similarity dimension. So this is capacity. And the definition of capacity is that the dimension equals logarithm as the size goes to 0 of log of the number of balls of radius r divided by log of 1 over r. And let me now show you the relationship of this to the self-similarity dimension, which seems more intuitive. In the self-similarity dimension, we took m pieces. Remember, we cut the lines into m pieces. So m, in some ways, is kind of inversely proportional to r. That is, if we cut it in more pieces, it's like going to finer scale. So m is kind of like the inverse of the scale at which we're looking. And we cut the object and we found that there were n objects, and that's kind of like the number of balls needed to cover things, so that's kind of like n of r. So if I use these two relationships to replace in here, what we have here is that d, and for the moment I'll keep this r equals 0 limit, is really log n divided by log m. And if there was no limit here, then this would be rearranging this d log m equals log n, or killing the logs. It would be n goes like m raised to the d, which was exactly what we had before, as I can prove by flipping back to here. So this is what we had before, that n equals m to the d, and now that's what we have down here. So this whole expression that we had for the self-similarity dimension up here is really just rewriting the equation, for the capacity, is rewriting the equation from the self-similarity dimension, replacing the number of copies of the object by the balls needed to cover the object, and replacing the pieces that we cut it to by the scale, and then taking the limit as things go to 0. So I'm trying to show you that this equation really comes from the same equation as the self-similarity dimension. Now what we normally have, and again this is the difference between the mathematics, and in a way the mathematics is not quite right, the capacity dimension in a way says that if we plot the log of the number of balls needed to cover the object against log 1 over r over here, then minus this slope will be the dimension, the capacity dimension. And in a way, this is all we really need to define the dimension. So we don't really need the limit as this thing goes to 0. What we're really interested is in the slope. So what we're really interested in is in the slope of this, not really in the limit that epsilon goes to 0. Now these epsilon goes to 0 have been developed for other mathematical reasons, but in a way we don't need that. All we really need is the slope. So in principle the mathematics could be redeveloped around the slopes, not about the limits. But for other reasons people have taken the limits. When how we're really handling data, we really use the slopes and not the limits. So we're in this situation where the mathematics doesn't match what we do in the analysis. And it's important to keep this in mind. And this works both ways. The mathematics can suggest how to do the measurement, and if we're doing something different, then maybe it would be useful to redevelop the mathematics in a different way that would match what we're doing. Okay, so, and I think this is important in general. We face this in physics and more so in biology, that we have mathematics that really isn't describing what we think it's describing, or is really different. We've got to be very careful about things. I think an example I mentioned before is when we have finite difference equations to represent a gas, we actually represent the gas as points of mass separated by little springs. And it's not exactly the same thing as a continuous gas. And this system can go into oscillations, for example, that a real system can't. And the equations sometimes produce oscillations, not because the equations are wrong, but because they represent a physical system that's different than the one we think we're trying to model. And so this is an important point to realize, that some of the mathematics is not what we really want, and we're using it anyway. So I've said this at least twice at some length, and I think it's an important point that's not usually understood, or is not usually expressed in the literature. Okay. So now we can go to the full Hausdorff-Besicovitch dimension. So this is what mathematicians usually refer to when they say dimension. And so this is dimension. Now this is very similar to the capacity. You can see here that so far we're setting up. In this case, we're not using balls. We can be more general. We'll just say that they're sets. And the reason why I want to say this is they could be sets of different size. I've drawn them all here as if they were sets of the same size. In fact, I've done a physical cut and paste from the previous one to here. So it's exactly the same figure. Except it actually didn't quite paste correctly the first time. Things fell behind each other and some of the lines in the circles disappeared. But that's been fixed. So basically we do the same deal. We look at some minimal covering of sets, and we'll see how that minimal covering changes as the size of the sets change. And in particular, what we're going to look at is something called a gauge function. So this looks pretty fancy. So let's try to do it in a less fancy way first. we're going to look at a function which I'm going to call h of s and r. And s is a parameter. I didn't notice that. S is a parameter that is going to be important, and r is the size at which we're looking at. And this function is going to be the sums over all the sets of the diameter of each set raised to the power s of the covering sets. Because each set can have a different diameter. This is a very, very technical mathematical definition, so I'm not going to go through the whole definition. I want to give you a flavor of the definition more than anything else. In terms of these sets, for example, we have to define what the diameter is. The diameter is kind of the largest distance between 2 points in the set, which is a reasonable definition, because these sets could be irregularly shaped, right? We're talking about very general things, right? And mathematicians have very open minds, so it can be very many different types of shapes to these covering sets. Formally, they're probably convex, but I'm not even sure of that. Okay. So this function, this is a useful thing, is called a gauge function. That is, what we're doing is we're taking something and raising it to some power. Like, we could have done anything to the diameter of these sets. We could have taken the logarithms of the diameters of the sets, or we could have taken the diameters of the sets and squared them, right? We could have done anything. This particular function of how we're handling the diameters of the sets by raising them to a power is called a gauge function. And the gauge function we're using here is to raise the sets to a power. Now, that's sort of right for what we're doing here, because everything else we've done so far involves powers. So somehow whatever we want to do here, it would make it reasonable to involve a power also. So that's the reason why we're choosing this gauge function. Now, what happens with h of s, if we go back here, is that when we take the limit as r goes to 0, as the size of the sets go to 0, this limit will either approach infinity or 0, depending on whether s is less than a certain value d, or s is greater than a certain value d. And then the d for which this happens is the dimension. Okay? So I'll say it again. In the limit as r goes to 0, this function h of s and r either approaches infinity or 0. And approaches infinity when s is less than d, and approaches 0 when s is greater than d. And the d that is this dividing line is the dimension. Now you notice that it's never s equals d, because this is a limiting process. We don't have to get to that value of d. We're either greater than that d or less than that d. So this is the usual idea of a limit. At one point I wrote that at s equals d, this has a certain behavior, and one of my mathematicians friends corrected me that we don't need to deal with s equals d. So this is a limit. We either approach it from above and below, and that defines d. Now let's see if the next one has some, no, it doesn't. Let me give you some understanding of what this means, because this looks very different than the dimensions we're looking at before. And let me show you that this is the same old self-similarity dimension, just hiding in a different disguise. So in the self-similarity dimension, the number of pieces will go like something like r to the minus d. So remember, we said the self-similarity dimension n goes like m to the d, and we said like m goes like 1 over r. So if I put both of these together, I get n goes like 1 over r to the d, or n goes like r to the minus d. Okay. So let's say I have something like a square, and we keep breaking it up into smaller pieces. Then the number of pieces will go like r to the minus 2. So let's say the covering sets are actually each of these little pieces. So the covering sets look something like this. And this is one way to generate a minimal covering of the object. So this function h of s of r will be the sum of the size of each of these sets, which will be something like r raised to the s power. Well, how many of these sets will there be? Well, we've just said that the number of sets will be like r to the minus 2. So our gauge function, this should be a little r, will then be the number of these sets, because we're summing over all the sets. So this sum will be, let me write it out, be like r over s plus r over s plus r over s as many times as we need. Well, for each of these separate sets, this one, this one, this one, this one. So this r is that one, this r is the second one, this r is the next one. So how many of these little boxes are? There are r to the minus 2 boxes. So the gauge function then is going to be like r to the minus 2, the number of these times the size of each one. Okay? Doing this better than I thought I would. So now we have h of s of r goes like r to the s minus 2. So if we get up to the next sheet of paper here, and we now take the limit as r goes to 0 of h of s of r, which is r to the s minus 2, what's going to happen? If s is greater than 2, this will be a positive power. As r goes to 0, h of s of r will go to 0. Okay? If s is less than 2, this is going to be like 1 over 0, and so h of s r will go to infinity. So the turnover here is 2. The Hausdorff-Besicovitch of the dimension of this plane, which is sitting here, is 2. So this area then has a Hausdorff-Besicovitch dimension of 2. So you can see that this sort of screwy procedure of taking this limit has just given us the same thing. Now this is a little bit different than the capacity. In a way it's really similar because we're covering the object in this case with these sets, and then we're counting the number of sets. In the capacity we counted them explicitly. We just looked at how the number of sets changed as the scale changed. Here we're doing this gauge function, and this has some more general properties, and in some ways is nicer and can handle more cases than the capacity. In general, for most of the cases we'll be dealing with, the capacity is pretty much equal to this Hausdorff-Besicovitch dimension, but it doesn't have to be equal. There are cases, for example, where the capacity is not equal to the Hausdorff-Besicovitch dimension. There are mathematical fractals that have some unusual properties where this is not the case. Now there's a formal relationship, which I don't remember offhand, which says that one of these is always greater than or equal to the other one, but I don't remember which one is which for the moment. So there is a relationship between the capacity and the Hausdorff-Besicovitch dimension, where one is always greater than or equal to the other one, but I don't remember that. For most of the cases we'll deal with in analyzing data, these two dimensions will be equal. So in most of the real cases involving data, these dimensions are equal. Now they won't be equal when we actually evaluate them from data, because the errors involved in evaluating each method will cause a little bit of a different problem. So quantitatively, when we evaluate them, there'll be a little bit different numbers. But formally, they're equal. Okay. So those are examples of 3 different fractal dimensions: the self-similarity dimension, which requires self-similarity, and these 2 more generalized dimensions, the Hausdorff-Besicovitch dimension and the fractal dimension. Maybe we should take a break here until 10:30. This looks like a good time to take a break because we're doing a lot of mathematics. And then we'll come back at 10:30 and then continue with dimension and statistical properties.